start our second session. The chair of this second session will be Professor Hyun-Sung Lee from the Seoul National University. Professor Lee, please.
Thank you. I'm very happy and honored to be here to give a talk on our recent work on organoids. So I have given a very big, long title, but actually it would be combining some of the genomic analysis that we have done from the pancreatic cancer patients by culturing the organoids and some of our advances in developing organoids for the precision medicine platform. Right, so my talk is composed of 3 parts. So for the first part, I'll be talking on our development of the pancreas organoids to reveal its identity on the genomics and also in the cellular identity as well. Right, so basically, everybody would be aware of this fact, but pardon me for reiterating this. So basically, when a patient goes into the hospital, if he or she has some problem in the pancreas, they'll be taking the fine needle biopsy. Unfortunately, 90% of these patients, even though they are diagnosed with cancer, 90% of them cannot take surgery because it's found probably too late or in a very bad place to take the surgery. So that means that our knowledge on pancreatic cancer might be limited based on the surgical samples. So what we thought was, because all of the patients would take the endoscopy anyway, so if we can culture the cells from the fine needle biopsy, we would be in a position to analyze those 90% of the cancer patients who were not really analyzed. So for that, of course, the 2-dimensional monolayer culture was not possible, so this was why we got into this 3-dimensional organoid culture technology. So with a lot of trials and errors, we were able to generate the 3D organoids from the FMB samples. So what is good about these FMB samples, organoids from the FMB samples, is that first, that we can analyze any of the endoscopy-derived samples, so that means any of the patients could be analyzed further with this method. And of course, because these cells would be cultured in a prolonged stage, that we would be in a position to do anything in the laboratory to analyze them. So with that, we first collaborated with Park Ju-kyung from Samsung Medical Center. She was formerly in Seoul National University Hospital here. So we had a wonderful collaboration that should give us any of the endoscopic samples in the morning and then deliver just directly into our laboratory in Gwanak in Seoul National University. So with that, we had a lot of the trials, and now we are in a position to have the success of maybe like 90% of the success rate with the fine needle biopsy derived organoid cultures. So what we did first is to actually make the DNA to see if we can detect any of the interesting mutations from there. For that, so this is the data to show the comparison of the cellularity of the FNB samples versus patient-derived organoids, which I'll call PDOs from now on. So as you can see from here, the cellularity of the FNB are very bad, as you know very well, because it's a very small aliquots, and of course they contain a lot of different things, so cells are not really there. But by culturing the organoids from these FMB samples, the data is actually from 2 weeks that we have a very good cellularity. So with that, as you can see here, when you compare the same patient sample from FNB versus PDO, you can see that a lot of these interesting mutations that we find from cancers were actually seen in the PDOs, but not really in the FNB. So this tells you that any of the analysis at the genomic level with the patient-derived organoids will give you much more information. So we were quite happy with this. So this is actually one of the summary results that we got, showing 12 different patient samples. Here you can see that at the left-hand side, a lot of these mutations, which actually we analyzed 50 of the cancer mutations. You can see the mutations were pretty much apparent in the PDOs, which were not really found from the FMB. So the sequencing data from the fine needle biopsy endoscopic samples in the hospital really does not really give you any idea of what these genomic profiles are. So with this, we went into a little bit deeper into looking into the genomic profiles. I have just exemplified here 4 mutations that we found, 4 mutations out of the 50. You can see that as KRAS mutation is actually one of the prominent mutations in pancreatic cancer. You can see that here in the pancreatic ductal adenocarcinoma region, which is diagnosed in a pathology lab, that they were all having 30% of them, no, 50% of them actually had KRAS mutations, and the mutations were like G12V or G12D, the famous oncogene mutations. Most prominent mutations were actually found from the P53, so 70% of the pancreatic cancers actually showed mutations in P53, and all of these were the famous hotspots like 273, 154, or like that. And interestingly, CDKN2A, which is also implicated in tumorigenesis, it was mutated here too. One of the interesting things was that this PDO165, derived from the 165 patient, actually was diagnosed as pancreatic, not pancreatic cancer, but pancreatitis, chronic pancreatitis. But it did show a very similar genomic profile compared to the adjacent 117, which is definitely a cancer patient. So we were quite puzzled with that. First we examined whether it was a contamination, but there was no contamination whatsoever. And they actually exhibited very similar mutation profiles. But compared to that, these 3, which come from autoimmune pancreatitis, did not really actually give this type of KRAS P53 mutation together. So one of the lessons that we learned here is that, of course, it was illustrated before, but a lot of the pancreatic cancer patients exhibit both mutations in KRAS and P53. So this was the most prominent one. And those which were not diagnosed as cancer did not show these mutations, but maybe some of the SMAD2 or other mutations too. This one is interesting, that PDO131, which was diagnosed as autoimmune pancreatitis, but it did have some, it did have one missense mutations in KRAS, but not in G12 area, but other region. Okay, so next thing what we found was interesting was on TGF-beta receptor 2 mutation. Here, remember these PDAC mutations, there we found like 30% of them having TGF-beta receptor 2 mutations. But interestingly, in the PDOs from the chronic autoimmune pancreatitis, they did not have the TGF-beta receptor 2 mutation, but maybe some of the downstream ones like SMAD2, SMAD4 were there. But basically, TGF-beta receptor 2 mutations were only found from these patients which were originally diagnosed as cancer. But again, here, interesting thing was that PDO165, originally pathologically diagnosed as chronic pancreatitis, also exhibited mutations in TGF-beta receptor 2. So it was quite interesting. So this could actually hint that 165 patient maybe could also develop pancreatic cancer, not just the inflammation. So this was one of the indications that organoid sequencing could give you some of the very early diagnosis. What happens is probably that 1 or 2 of the tumor cells were actually growing out in this organoid culture. So that was one of the things we thought. But of course, the pathological examination was still pancreatitis. Well, the reason that we focus on TGF-beta receptor 2 mutation is because of some of the reports generated. As you can see here, that the overall survival is compared between the TGF-beta receptor 2 mutation and those cases without TGF-beta receptor mutations. And these all include KRAS and P53 mutations altogether. On that, what we see is that TGF-beta receptor mutation has actually lower overall survival. So it could mean that those patients who are having TGF-beta receptor 2 mutations should be treated differently or maybe in a different way to target this pathway. There are some reports that suppressing this pathway actually alleviates some of the cancer cells. Right, anyway, coming back to this story of 165 patient, what we saw was that, this patient had KRAS mutated, P53 mutated, and even TGF-beta receptor 2 mutated, showing that if they grew out, that this would have a very bad prognostics. But basically, what we found was that these two genes, CFTR and CTRC, which are only implicated in pancreatitis in the fibroblast, is only found from the 165. So what we think is that by expanding the epithelial cell, pancreatic ductal epithelial cells in this organoid culture, actually gave some the information which could never be found from the FMB, that maybe very rare cancer cells were growing out in this organoid culture. So that means that yes, these are in pathology, yes, they are pancreatitis with a lot of the fibroblast because they have these signatures like CFTR, CTRC, but we were able to detect a very early stage of tumor. That is what we interpret out of this data. So so now what we are doing is that we are trying to have more of these cases to kind of confirm whether this organoid sequencing could actually overcome the hurdle of late diagnosis of pancreatic cancer and show any hope for those who would be having just very early stage of the rare cancer cells. Right, so of course the beauty of these organoid cultures is that you not only sequence these, but simultaneously we can culture them and examine in a different way, such as immunofluorescence. Here, the thing, the PD-165, which I'm repeatedly talking about, which was pathologically diagnosed as chronic pancreatitis, but features mutations just like 117 cancer patient. What we saw was that in the cell biology, the staining pattern between 117 and 165 were quite different, which means that these cell biological analysis simultaneously done with the mutation profiles would give you the idea of which therapy or choice of therapy should be taken if the surgery is not possible. So we were quite happy about these results. So it did actually have very different pattern. In the genetic profile was similar, but cellular patterns were quite different. So maybe these are the reasons why 165 were diagnosed differently. Right, so other things that we found was that when we did the karyotypings from this organoid culture, so the beauty of this organoid culture is because you can culture out the cell aggregates from very rare cells, that you can do anything that you want. So my lab is actually expertised in mitosis and telomeres and tumor suppressors. So one of the things that we actually do very well is the karyotypings, because when you examine the metaphase chromosomes, you learn many things out of that. Interestingly, what we found was that this was never reported, so it's a novel information that we have, that in the PD-117, which is a cancer patient, we found that they had PMSCs. So PMSC stands for premature cystochromatid breakage, which is a feature of like premature mitosis segregation, and also some of the coisins are involved, coisin mutations are involved in these PMSCs, and a lot of somatic cancers have this PMSC, but it was never reported from pancreatic cancer before. But with analysis in the FMB-derived organoids, we found that a majority of the pancreatic cancers would have like mitotic instability as well, manifested by this PMSC. So this would be one of the information that we could get out of organoid culture. So altogether, these could combine comprehensive information would actually give you more idea of what is really going on in pancreatic tumorigenesis. On top of that, so because we were able to culture them in a standardized, robust way, we asked if we could actually kind of discriminate one patient from the other or help which drug would be the best for that specific person. Well, so this would be one of the preliminary stage of doing some of the experiments, but this is actually for the precision medicine. So if the person cannot take the surgery, what drug should be given to this patient? So for that, there are only 5 drugs which are approved for treating pancreatic cancer. So this is one of the example. So with the collaboration with Bundang Hospital, Bundang Seoul National University Hospital, we took surgical samples. These are the surgical samples. We took surgical samples in the company Nexten Bio, the startup that I'm one of the co-founder, and they cultured the organoids from them and treated with the famous gemcitabine. And you can see that all these patient organoids behave differently in response to gemcitabine. Next, the same organoids were treated with 5-FU, another famous drug, and the patterns are quite very dramatically different. So these are the examples that we can culture the organoids on a kind of a replica plate, and these drug responses could be tested simultaneously in a different level. So this would be one of the way to go for that, before really treating the patient, if organoids could be cultured out from the FMB, that you might be able to suggest for the best drug possible, or even do the combinatorial, because these are done in the live cells. So basically you can treat in a sequential way. Right, so with this, so we are hoping that we can develop a new platform to apply into precision medicine. So one is that we do multi-omics on these organoids, so to learn any of the information that we have. But simultaneously, we do a lot of the drug screenings on a standardized chip. And also we integrate all of the informations together. So if the data was big enough to do this repeatedly, we are hoping that we are doing it now anyway. So we are putting into the machine learning. So hoping to kind of detect or develop a few novel biomarkers for that. So without repeating all of this for every single patient, we will be in a position to suggest for a right drug for the patient. So this is probably the way to go for, and my lab is working hard on this development. Okay, for the next thing, so to change the gears, I'm going to show you how you can target the specific pathway. So from the second part, this is not the human cancer, but actually the mouse, genetically modified mouse. So we are doing a lot of study on the tumor suppressor BRCA2, and with the study, we have actually seen that BRCA2 has some roles in mitosis, a very critical role, and it's actually working as a platform for mitotic checkpoint proteins to come together, and particularly the checkpoint protein BABA1 is the core protein which interacts with BRCA2, and on this BRCA2 platform, it is modified based on acetylation, deacetylation, and also phosphorylations. So it's a scaffold protein, but it is essential in regulating progression of the mitosis. So finding this, what we did was, because we had this BRCA2 conditional knockout, we actually developed the pancreatic organoids from this mouse, and we treated the cells with various drugs which are involved in HDAC inhibition. And what we found was that because of the pathway for BABA1, what we found was that with these treatments, we could actually see that there was a huge difference, that the pan-HDAC inhibitor for the proteins were actually not killing BRCA2 negative cells, but organoids, but this epigenetic drug was quite useful for treating BRCA2 mutated pancreatic organoids. So this was one of the examples that to see that this type of organoid-based screening could work for targeting specific pathways. So this was one example. And the next thing is that a new type of cancer called alternative lengthening of telomeres is one of the things that we study in the lab. And again, we found that BRCA2 mutated cells could undergo the ALT inductions. So because these ALT cancers are not really studied that much, that the targeting drug is not there yet. So what we did was we developed a model of ALT cancers and hoping to kind of find the right drug or even screen for it to target the BRCA2 deficient ALT cancers. So these were quite working well. So we are quite, I mean, maybe next time I'll be in a position to show you one of the examples for targeting ALT cancers. So for this, my experiences are that these organoids are actually much better compared to the monolayer cancer cell cultures in targeting a specific pathway or even giving the ex vivo phenotype of the patient. Right, so lastly, I'll show you some of the development that we have done in collaboration with the startup NextGen Bio in making the standardized robust microfluidic chip. The reason is this, like in the organoid cultures, you basically have the ECM, the extracellular matrix, which is the matrigel. On the matrigel, there are three problems. One is that matrigel comes from the sarcomas of the mouse, so they are actually not really approved. If you do any of the drug screenings on the matrigel, it would not be approved by the FDA. That's one problem. And because we were quite interested in doing some of the drug screening, so that was one of the hurdles. Secondly, the size of the organoids would actually affect the response to each drug, and that's actually not possible in the matrigel culture. So for that, we wanted to develop a matrigel-free system. Not exactly 0%, but 1% of the matrigel is included here, so mostly free. Matrigel free, and also we wanted to control the size of the organoid or the number of the cells in this aggregate. And for that, we, in collaboration with Dr. Jung Seok from Korea University, Mechanical Engineering, and also with NextGen Bio, we developed a honeycomb-like microfluidic chip. Here you can see that the size of the organoids would be controlled, so they are in a very regular, standardized chip. So with this, what we did was, of course, first we confirmed that the genomic features were all the same compared to the matrigel cultured organoid. And secondly, we compared that they are growing in a very good position, like they can be cultured for 3 months without any problem with the right successful subcultures. With that, this is one of the examples. So these are the human cancer human pancreas cancer organoids again. Here we put them into this honeycomb microfluidic chip that we developed, and these are actually the drugs which was suggested for a study that this works on this pancreatic cancer. We saw that actually the assay to do any of the MTT or the quantitative assays were working much better in this microfluidic chip. On top of that, the use of the drugs were reduced to 1/10-fold compared to the matrigel base, because the drugs would be absorbed into these cells much better in this microfluidic chip. So we have some success in this development of this drug screening. So with this, we are aiming to do a lot of the other studies. And also, nowadays, we do the genomic profiling on this microfluidic chip, because you can get rid of the matrigel, so that's much better and cleaner. Right, so other things that we do is that because these organoids can be imaged in live, that we do a lot of studies. And one of the things is that looking into the mitosis and how they behave. And of course, you can see how they divide. And this is the H2B GFP organoid, that you can see that these aggregates are dividing, and actually you can take the live images for more than 48 hours, of course on the microfluidic chip, but not in the matrigel, because we don't want to get that much of the problem with the matrigel. So with this, we are hoping to develop some other methods to study organoid in a more physiological way. Right, so all of these could be combined and applied into industry. So the startup company, Nexten Bio, which I'm a co-founder of, is actually developing this microfluidic chip-based organoid cultures, and of course they can do a lot of different assays. So now they have like 13 different organoids cultured, and some of them are actually paired, meaning that normal organoid versus cancer organoids from the same cancer patient. So those are the valuable samples that they are actually doing, a lot of the bankings and doing a lot of the screenings with the drugs. Okay, so these are the things, but of course there are some caveats, like, you know, these are really growing very well, but basically this is not actually like this real organ. So what you can do with that, we are nowadays doing some of the co-culture system to provide like vascular system and the fibroblast and also the immune cells, because this is a three-dimensional culture system. So it can be done in that way. So we find that, although it's a preliminary stage, that we find that some of the genomic features change based on co-culture. So that gives you that we are able to kind of constitute the microenvironment for the cancer. So if that's possible, then if we apply that into the microfluidic chip, so we will be able to screen the right drug for the patient with the microenvironment provided. Okay, this is all, and I'd like to thank all the members of the lab who gave all the ideas, but basically a lot of the work that I presented was done by Soo-hyun Lee, So-young-ju, and Ji-ho Park, who got PhD and is now in Nexten Bio. And also a lot of the help were coming from Samsung Medical Center, Park Ju-kyung. Also, the genomic analysis was done from Semin Lee from UNIST. And I'd like to thank especially to Sit Jung-seok from Korea University, who was a wonderful collaborator in making the microfluidic chip right for our research. Okay, with that, thank you very much, and I'd like to take any questions.
Thank you for your wonderful lecture. I have one question. So, as you know, the establishing organoid can grow from the cancer cell and also from this normal cell. So when you establish cancer organoid from the cancer tissue, how do you differentiate this cancer organoid or normal organoid?
Very good question. So right now we don't do that much. We don't put that much effort into discriminating normal cell versus cancer cell in that organoid yet. But basically, by subculturing, what we found was that if you change the media, the normal organoids would grow better, and in depriving some of the factors that cancer organoids grow better. So that's one of the things that you can do to discriminate normal organoid versus cancer organoid based on the media change. So some of the examples that I showed you in Nexten Bio, they have this paired system based on that method, differentiating the culture media. We are hoping to kind of develop a smarter way to barcode them, because it is always useful to have normal versus cancer, not only just the cancer. So this is kind of very labor-intensive way to do the discrimination by culturing them. But if we develop some other ways, we are hoping to do some kind of less labor-intensive way to discriminate them. So yes, it's on the way, but not still there yet. Thank you very much.
Yeah, thank you for a very interesting talk. I'm Uchan Lee from GMI. On your last topic, you said about the microplate, Yeah. So, as you know, Toshiro Sato or next speaker Hans Kleber said that we need matrigel to not just sustain the structure, but like a basal membrane. I mean
Yeah.
the junction, and
Yeah.
we need laminin. But so my question is, do you know why we can culture organoid in just that
Fluid chip.
fluid? Yeah.
Yes. Wonderful question. So basically, this fluidic chip is based on provided to give the right force and strength. So what extracellular matrix like laminin, elastin, a lot of these collagens are doing is actually giving the force and interactions. So those could be provided if you design in a fluidic chip in a right material, and also the degrees and everything, like angles and everything. So that's what is about this microfluidic chip. We found that, interestingly, when we did kind of the optimizations, we found that these chips were as good as Matrigel. So that has some of the advantages, because Matrigels are sometimes really pain, because you have to get rid of them to do the genomic profiling and also RNA sequencing, and you don't want the contaminated DNA from the Matrigel. So I'm quite happy with this. Secondly, we are developing the co-culture system. So for there, you can culture with fibroblasts, which means that the secreted things from the fibroblasts would also give you some right forces, and also the factors which would be coming from the ECM as well, so the dermis. So I think that's why. So the right force, not the factors, but actually the interactions between the cells and also with the fluidic chip. Okay, yes.
Yeah, thank you for an interesting talk. And I'm from Gyeongbuk National University. And my question is a little bit similar to Professor Kim, but a little bit different direction. And in your patient-derived like organoids, such as cancers, and within those cancers, there are many heterogeneous diversity. And I guess like some kind of organoid culture can selectively or preferentially select a certain population. And then did you have any experience and share the comments of that things?
Yeah, it's very good comment and very good question. So we were aware of that from the beginning. So at first we didn't have much methods to discriminate that. So we actually have done, we are planning to do kind of sequential stainings, like whether there are mutations selected out. But right now what we found was that like even if we did the sequencing in 3 weeks or even 2 months after, that you can have a lot more other noises, but basically the prominent mutations are still there. So we are not still, we don't have the right method yet to discriminate which are the noises or which are really kind of selecting out. But basically in cancer biology, we do learn of these important genes, but anyway, interestingly, they are still maintained. So it's actually reflecting the prominent features of the patient. But yes, there's a caveat that in culture, it might be selecting out for some other cell types compared to others. For that, now we are doing single-cell RNA-seq to see if there are the heterogeneity actually changes in prolonged culture. So I don't have the right answer to that yet, but we are trying to kind of reveal or nail down the question. Yes, there are some heterogeneity. We did some 8 of the organoid cultures, and the preliminary single-cell RNA gene analysis tells you that the heterogeneity is there, but it's not as much as the tissue, of course.